Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast with Richard Jacobs.
Before you get into this audio, I just need to make the disclaimer that the interviewee that I speak to, Jake Becraft, at the end of the podcast, he recommends that, I guess, everyone pretty much get the Moderna mRNA vaccine for COVID-19. I have to make the disclaimer that we here at the podcast do not make the same assertion. That is the view of Jake Becraft alone. And we're not here to give medical advice to people to tell them to either vaccinate or not vaccinate. We're here to stay out of that debate. Jake, again, has chosen to make that assertion on this podcast. But I just want to let you know that his views are his views, not right or wrong, but they are not the views of Finding Genius or Finding Genius Foundation. With that being said, please listen in. Thank you. Hello, this is Richard Jacobs with the Finding Genius podcast, now part of the Finding Genius Foundation. Quick note is that our new book on understanding viruses is now on Amazon and Kindle and soon to be Audible. To find it, go to Amazon and type in Finding Genius and you'll see the book. And I think it'll be very informative for those interested. So today, my guest is Jake Becraft. He's the CEO of Strand Therapeutics. And we're going to talk about programmable mRNA therapeutics for cancer. So, Jake, thanks for coming.
Thanks for having me, Rich.
Yeah, if you would, tell me a bit about your background. And then I don't know if you started Strand or just came in to be the CEO, but then I want to hear about what Strand does.
Yeah, no, absolutely. So, Strand is a company I co-founded along with my PhD advisor, coworker, another postdoc in the lab, and another professor from MIT. So maybe to walk it back, my background, I've spent the past, more than a decade working in what I consider the field of therapeutic gene therapy or therapeutic gene transfer, right? Finding ways that we can modulate disease by creating systems that are capable of delivering genes into cells. So what this allows us to do, either with a virus or synthetic DNA or synthetic mRNA, is kind of treat the core of a disease, right? Almost all non-infectious disease have some genetic component to them, and that could be, something as intensely understood like cystic fibrosis, having a genetic mutation, or something like cancer that has a whole number of genetic mutations that cause the dysregulation. And so I worked in this field. I started in this research space at the University of Illinois, working on delivery technologies. I came out to MIT in the fall of 2013 to work on more, I think, engineered ways of controlling the genes that we would deliver. So I honed in on a field called synthetic biology. And synthetic biology essentially allows us to create gene circuits or programmable nucleic acid sequences, allow us to make DNA or messenger RNA that can perform in new and exciting ways. Things like nucleic acids that only activate when they're in a diseased tissue, for example. And I began working on that field, that idea of gene circuits and programmability with Ron Weiss, who's the director of the MIT Synthetic Biology Center, and a postdoc in the lab who I was working closely with named Tasuku Kitada. He came to me and basically was talking about how there was this new area of research called messenger RNA therapeutics. Moderna was just getting their start. They were just starting to grow. They were probably worth about $100 million at the time as a new and exciting startup. And Tasuku's pitch to me was the future is going to be based on messenger RNA. And if this is true, it will need new programmability technologies, because while synthetic biology had worked a lot on DNA-based approaches and different sorts of DNA circuitry, there was almost nothing that existed for mRNA. And so we started working on that. About 6 years later, that technology was very mature and mRNA had really grown in popularity. And so in 2018, we spun the technology out of MIT, myself and Tasuku being the two co-founders of the company, and Ron Weiss and Daryl Irvine at the MIT Koch Institute being the scientific founders, and have been growing this company ever since then.
For mRNA to target diseased tissue or diseased cells, how do you know what cells to target, and how do you package the mRNA so it targets the proper cells versus not?
Yeah, so this idea of targeting cells has been around for a while, right? Like from a molecular biology point of view, a lot of disease we now completely understand what the genetic markers of the disease are, and also what the genetic dysfunctions are. The Human Genome Project has, over the past 20 years, had just a wealth of data for us to kind of pull from. So that's been a very exciting aspect. I guess where we end up kind of having an issue with how we develop therapeutics is now, once we understand what's wrong, how do we get into the correct cell type with the correct specificity, right? And so there are any number of biomarkers that exist on inside or outside of a cell, given a given disease state. And so what the real key is building what we call genetic sensors. So these are things that are encoded on a nucleic acid that are able to sense a biological molecule in some sort of way. And then biological circuitry basically is able to take the result of the sensor's output at the genetic level and process that in order to turn on or turn off whatever sort of genes we are encoding on our vectors, so that when these things go into a cell, they will respond to whatever is inside of the cell, and the therapy itself will be able to make a sort of logic-based decision through like AND gates and OR gates, how like early computers worked. Use that decision-making process to decide whether or not to output the therapeutic that we've encoded on the vector itself.
Well, what is the mechanism? I mean, is it packaged inside of a lipid membrane or a nanoparticle, or what's?
Yeah. So
how do you deliver the mRNA?
Yeah, so the mRNAs that we use are, as of now, all delivered with lipid nanoparticles. That's kind of the cutting edge of the science, though there's a lot of new and exciting delivery technologies that we're also interested in pursuing. But lipid nanoparticles are really having a time in the spotlight right now, obviously, with the COVID vaccines. But the technology underpinning lipid nanoparticle technology is just wildly exciting. And so that's what we utilize. But all of the programming, all of the logic, all of the so-called smart therapeutics aspects of this are all encoded on the sequence of messenger RNA itself. And so we build very complex mRNA molecules that it's the key to the innovation is all based off the sequence of the messenger RNA.
So how does the mRNA selectively enter into the right cells? Like, what surface features does it interact with on the right cells, and what surface features would it not interact with on the wrong cells?
Right. So this is a fantastic question, and really the way that people have been trying to solve the issue of specificity for the past, couple of decades has been to target surface markers to get the RNA into only the correct cells. Where Strand takes a difference in our approach is that we don't care so much in the selectivity of uptake, because the messenger RNA themselves is going to be engineered that once it is inside of the cell, it will only activate and actuate its therapeutic payload if this cell is the correct sort of recipient cell. So what we need from the delivery vehicles is really just to get us into the correct tissue. It doesn't have to only get into the correct tissue. There can be heterogeneity in the types of cells that it's delivered to, as long as we do have a significant portion getting into a given target tissue. And then we program the messenger RNA itself such that it will only activate when it's in that final destination. I like to think about it kind of as the lipid nanoparticle will get you to the right neighborhood, and then we utilize the mRNA logic and the mRNA programming to get us into the correct house.
Well, what does the mRNA programming look like? Like, what's an example of how that would work once it's inside of a cell?
Right. So some of the papers that we published on this when I was at MIT outline one thing that we call a classifier circuit. And so what a classifier circuit does is, inside of our cells are these short RNAs. So they're not messenger RNAs. They're short RNAs called silencing RNAs. And so those silencing RNAs exist inside of different cells, and they are also expressed at different sorts of levels depending on the type of cell that they're inside of. For example, in some cancers, they will overexpress certain silencing RNAs, and those silencing RNAs go on to disrupt the cell's ability to control its growth, for instance. And what you can actually do is you can take those silencing RNAs and create sensors on the RNA sequence itself.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
Such that when the mRNA enters a given cell type, it will bind or react to certain silencing RNAs, whether they're there or whether they're present or not. And that differential expression is something that you can use, known microRNA expression patterns, feed that pattern into a circuit design algorithm, and actually output a sequence of messenger RNA that will only actuate itself in the presence or absence of certain microRNA species. And that allows you this sort of specificity that is just unable to be attained in a lot of other sort of design paradigms.
So the messenger RNA will encode the instruction of certain proteins to be expressed more than they would be, or enzymes, or what's the end effect of the mRNA influencing the cell?
That's really the beauty of messenger RNA, right? Is that, anything can be encoded on the protein, right? And so in the case of the drugs that we're developing for solid tumors, for instance, the protein that's actuated is a immunostimulatory protein. So it will go out and stimulate an immune response against the tumor and really aid in that sort of immune response that a lot of new immunotherapy drugs are really trying to get at the heart of, right? But in the case of other drugs, right, it could replace a transmembrane protein, a receptor, a sort of ion channel. It could go in and express the sky's the limit, right? Something intracellular. You could even express CRISPR off of these RNAs and use that to edit the genome. All of it is possible. The fundamental technology is just an ability to have more control over the messenger RNA molecule itself. And messenger RNA is really just a data carrier. It's a data carrying molecule that's transient in nature, which also makes it safer than something like a DNA or a virus.
It doesn't have to be the specific application you're working on, but again, what would be the end result? Would it upregulate the expression or production of certain proteins or enzymes, or what are some of the things that could be done, and why would that be useful?
So you can express off of the messenger RNA really any protein, right? And so in case of immuno-oncology, right, you express a protein that engages with the immune system. In the case of a rare disease like cystic fibrosis, you would express the protein that is mutated, that causes the disease itself, and replace it with the correct copy. I guess the answer is that the sky is really the limit on what you can do with messenger RNA and what that protein does itself. It's not necessarily about stimulating the cell to produce an enzyme or a protein in a certain way. You can just directly encode the enzyme or the protein itself onto the messenger RNA carrier and have that expressed from the vector directly. You can also, of course, have something like a transcription factor that would then go and activate the genome and activate the expression of a different protein if that's something that you wanted to do as well. But truly, mRNA is such a versatile molecule that it allows us to really follow the science and follow, what is needed in order to fix this underlying genetic condition.
Well, what's the residency time of it? Does it, once it enters into a cell, is there enough of it that'll just keep acting forever in the fashion you wish to, or do you have to replenish it and continually give doses over a period of time?
Yeah, messenger RNA is a very transient molecule. And so, for instance, the COVID vaccines that use technology called modified RNA, those will only express for a couple of days at most, right? 2 or 3 days. And then the mRNA is degraded because it's in nature, your DNA is using messenger RNA to send its gene signals out of the nucleus into the cytoplasm where the genes are then converted into proteins. And that sort of signaling is supposed to be transient, as you need to be able to turn your genes on and off. It's the same with delivering these synthetic messenger RNAs. Now,
if you like this podcast, please click the link in the description to subscribe and review us on iTunes.
That being said, the specific technology that we've developed at MIT and now at Strand and are really pioneering allows us the ability to express for much longer than that. So usually we look at timescales on the matter of weeks. So anywhere from 2 to 4 or 5 weeks of expression. And again, this is very advantageous from a safety perspective, right? You're not making a genetic or a lasting change in a patient that will have, yearly sort of consequences. You're making a short-term sort of delivery there, but then you have this ability to redose into the patient, to give another dose. And depending on the disease that you're looking at, it can, what you want in terms of expression duration can vary.
How do you know that the mRNA is going to degrade and how fast it degrades? I mean, how can you tell? Are there biomarkers that you evaluate to show its degradation, or, what happens?
Yeah. mRNA is itself just a transient molecule. It will degrade over time. It does not have the staying power of other sorts of molecules. It's a well-known biochemical phenomenon. But of course, we've done all sorts of studies in everything from cells in a petri dish into actual larger mammals. And you see that, over time there is an expression kind of wax and waning, but that does kind of subside, even in a number of weeks. It's usually that you need to take the approach of getting the expression up rather than kind of the opposite of getting the expression to be shorter.
Oh, once you give someone a dose of mRNA, I know it depends what for, but, as an example, a cancer example, or, I don't know, a cystic fibrosis example, like what has been shown clinically once people get a dose, or if it's just in animals, a dose of mRNA, like what are some of the effects it's had on cells and on that organism?
The biggest kind of challenge of messenger RNA therapeutics is usually immunogenicity. So a lot of viruses utilize mRNA, right? COVID is an RNA virus. HIV is a retrovirus, so it's made of RNA and turns itself into DNA in the body. And so your cells have evolved these sensors basically that try to sense for when there is exogenous RNA added to the system, and they use that as a kind of a marker of a potential viral infection. And the cells have what's called the innate immune system, and that will mount this offense against inside the cell whenever it thinks that maybe there's a virus that's gotten inside of it. So that's been the major challenge, and that's when we talk about modified RNA, that usually refers to some modifications that were discovered and developed by a woman named Katie Carrico and a man named Drew Weissman at UPenn in I believe it was around 2007. Those advances in the modified RNA field those advances in the RNA field allowed us to create less immunogenic mRNAs and really underpin the technology that we use today for something like the COVID vaccines. It's continually a sort of a design challenge, right? Where the longer expression that you want to have of the mRNA, the more of a risk of these sorts of immune responses comes out. So it's something that I think it takes a lot of work to design around. That's really where, if we want to dive deep into the scientific underpinnings and some of the things we've published in the messenger RNA space, you can see that there are all sorts of creative bioengineering solutions to get messenger RNAs to behave better as a real therapeutic substance, but where rolling that out is just a question of intense bioengineering. You have to really work on how you design these vectors.
What do you expect in the next year or two that you're going to be able to do? Like, how is any form of this yet commercialized, or like where are you guys at on the trajectory?
Yeah, we're an earlier stage biotech company. We were founded in 2018. So for instance, like Moderna and BioNTech have been around since the 2000s. Biotech companies become commercial very slowly, but our right now we'll be doing our first human clinical trials next year in 2022. That'll be in the solid tumor immuno-oncology space, and begin treating patients there. And really, then it's a matter of scaling out the platform, right? Building better drugs and continuing to attack new disease areas for the foreseeable future. So we are early, but growing rapidly, and really, really excited about the places that this technology could be applied.
Okay. Again, you see it for cancer, or for what areas specifically have the most need, and what areas are you going to be targeting this technology to first, you believe?
Yeah, our clinical trial next year will be focused on cancer, specifically solid tumors. So looking at areas like breast cancer, melanoma, head and neck cancers. From there, we'll be growing. It will be growing the programs in our pipeline, the different drugs that we're developing, as this modality is certainly incredibly diverse. And so our goal is to really make that diversity a cornerstone of our engineering, and be able to deploy it in other areas like rare disease, cell therapies, all sorts of other areas where we think mRNA can really be transformational.
And what's the best way for people to keep tabs on Strand? Where can they go?
They can follow our Twitter and our LinkedIn pages, is where we're the most active. They can follow my Twitter and my LinkedIn, as well, as I obviously talk a lot about what the company is doing. And go to our website and just kind of keep tabs on the different announcements we're making.
Okay. Anything else that you want me to mention that we haven't talked about?
Yeah, I mean, I don't know if I have anything specific. I think everyone should get vaccinated, depending on when this podcast comes out. Everyone should go get their mRNA vaccines. I think getting vaccinated is a huge step for us forward in terms of a return to normalcy, or some form of normalcy, and that the mRNA vaccines are, while the technology is very new to the general public, this is something that the field of biotech and bioengineering have been working on for the better part of 2 decades. And so while it might be new in the public conscious, this is technology that we have been diligently developing for many, many years. In addition to that, I think it's been interesting to see the increased public attention of biotech and biotech investing among the more general public. Biotech has been very isolated as an investment class for basically its entire history, because it is very complex. And so I'm just very excited to see more people taking an interest in these deep science and exciting science opportunities. And so I think that companies like Strand are ones people should keep tabs on for when they may have an opportunity to invest in the company as well.
Very good. Well, Jake, thanks for coming on the podcast. I appreciate it.
Thanks a lot, Rich. I appreciate you having me.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.